How do amoeboid cells move? We'll talk about two general mechanisms. The first of those mechanisms involves myosin and actin, microfilaments of actin, and myosin that's attached to other myosins and to the cytoskeleton. When something activates the myosin and the actin, which we'll talk about in a bit, the myosin heads attach and go through the power stroke and moves this entire strand of actin, putting force on the leading edge to expand. So both of these are putting pressure on the leading edge and cause expansion. So what actually triggers this to start? There are multiple mechanisms. One of the major ones involves tropomyosin, a binding protein that covers the binding sites on actin where myosin can bind. So when those are covered, a myosin head cannot bind. There can be no interaction. Attached to the tropomyosin is another protein, troponin, a calcium binding protein, and calciums bind, and when they bind, they change conformation, and that change in conformation moves the tropomyosin off of the binding sites. Once the tropomyosin is off the binding sites, a myosin head can bind and go through the power stroke and move this microfilament myosin, again putting pressure on the leading edge of the membrane or pulling actin away from the trailing edge. The second mechanism involves microtubules that are spreading out from the microtubule organizing center, and at the back end of the cell there's disassembly of actin microfilaments of cytoskeleton. So proteins are activated that break up these and move them toward the nucleus in the microtubule organizing center. Dyneins move some of this material with cargo apparently toward the microtubule organizing center, toward the nucleus, and kinesins move material outward. Actin microfilaments and cytoskeleton elements are being removed at the trailing edge, and so our trailing edge is moving forward. And at the other edge, there are new actin microfilaments being formed. Proteins that can cause microfilaments to join to form a microfilament are activated, and that activation causes new microfilaments to be formed at the leading edge. And those microfilaments put pressure, exert force to move the membrane boundary forward and move the cell. So how can those be activated? In both cases, they can be activated by receptor systems and second messengers in various ways that activate the proteins to cause microfilament growth or to cause actin and myosin movement. So imagine that some secreted chemical or a food molecule binds to a receptor. That receptor binding activates a G protein to separate, going to adenylate cyclase, which converts ATP into cyclic AMP, and cyclic AMP then might bind to a protein that causes microfilament growth. And all these events are going on only in this part of the cell, and so only in this part of the cell is there microfilament growth. So this part of the cell will advance. Another possible mechanism is that the cyclic AMP can open a calcium channel, allowing in calcium, and remember the calcium can bind to troponin. Again, if the calcium is only in this advancing part of the cell, then actin and myosin will be interacting only here. So only in the area where the receptor is binding would you get microfilament growth and or actin and myosin movements. There are many different forms of myosin that go to specialized different regions of a cell and are used in different ways. There are different troponins There are multiple proteins that can cause microfilament growth, and so different kinds of cells are regulated and move in some different ways. The two ways I've just discussed are two of the major ones.